Let's take a look at how diet may play a role in evolutionary change by considering milk and lactose intolerance. Lactose intolerance is a condition in which an individual is unable to break down lactose, the sugar present in milk, and suffers gastronomical discomfort when exposed to it. Today, lactose intolerance is the most common protein deficiency in the world, affecting 65% of the world's adult population. For most of our evolutionary history, humans had no need to digest lactose in adulthood because milk was not part of our diets. This changed around 8,500 years ago, when humans began to domesticate livestock and subsisted on their secondary products, such as blood and milk. Lactose tolerance would have provided selective advantages for those who could tolerate it and have access to milk, including a clean source of water and a year-round source of fat, sugar, protein, and micronutrients. When people shifted their subsistence economies to pastoralism and dairying during the Neolithic, it placed a strong selective pressure on lactose persistence. Today, the dairying legacy is apparent when examining the distribution of lactase presence across the world. Lactose persistence is far more common among populations from Europe and East Africa, where dairying is common, than among populations from Asia, West Africa, America, and the Mediterranean, where dairying is rare. Means of identifying what early humans and hominids consumed can be direct, for example, stable isotope ratios, tooth wear, cavities, or indirect, for example, genetic adaptations, plants and animal remains, and pottery residues. Genes for lactose persistence are a long-term consequence of dairying, but are not a direct measurement of whether or not an individual actually consumed milk. Recently, researchers have discovered a direct indicator of human milk consumption in the past: the presence of milk-derived protein beta-lactoglobulin trapped in dental calculus, or mineralized plaque. For the first time, actual consumption of milk from cows, goats, or sheep can be positively identified on a person-to-person basis. Using this new method on human skeletal remains, researchers revealed that as early as 5,000 years ago, people in Eurasia, approximately 25% of the sample, consumed milk, whereas people in West Africa did not. Researchers also showed that milk consumption declined in the diets of Norse Greenland settlers between the 9th and 15th centuries AD, supporting the hypothesis that the settlers and their livestock starved after they failed to adequately adapt their lifestyles and subsistence to the harsh environment. The presence of traces of milk and dental calculus in Greenland inhabitants offers a window into cultural conservatism. The occasional instances where culture and tradition impair our abilities to survive and thrive when circumstances change. We think the first Neolithic farmers and herders were probably lactose intolerant based on scattered Neolithic DNA evidence. Lactase persistence likely evolved after people began to drink milk, which would make it a classic case of gene cultural evolution. In this scenario, there must have been untold scores of people who suffered either via mortality or reduced reproductive rates due to an inability to drink milk. Otherwise, the allele frequency would not have shifted in dairying populations. The ability to identify milk consumption among lactose-intolerant individuals shed new light on the people's experiences. We may also find that milk consumption commenced after lactase persistence evolved, in which case another explanation for the trait's origin needs to be developed. Direct indicators of diet help us better understand evolutionary changes and why it happens.